Hello and welcome back to my YouTube channel. Today we are going to see the State Board Class 8 Science chapter number 10, Cell and Cell Organelles, part 2. In this we had already said some of the cell organelles in the previous part, so we will continue with that. Today the first what we are going to see is the endoplasmic reticulum. This particular cell organelle is helping in conducting the various substances inside the cell. Means any particular material is supposed to be carried from one place to the another place inside the cell, and there is a role of the endoplasmic reticulum in conducting that. The structure consisting of interconnected miniature tubes and sheets filled with fluid. Endoplasmic reticulum is connected to nucleus from inner side to plasma membrane from outer side. The main functions of the ER, which we call as endoplasmic reticulum. The first one, it is a framework that supports cell. The next one is the conduction of the proteins inside the cell. Then the toxins that have entered the body through food, air and water are made water soluble by ER and then flushed out of the body. So this is one of the most important function what the ER is having. The next one is the Golgi complex. It is made up of 5 to 8 hollow flat sacs placed parallel to each other. These sacs are called as the cystine and are filled with different enzymes. The proteins coming from ER are enclosed in vesicles which come towards Golgi complex via cytoplasm. They fuse with the formation phase of the Golgi membrane and empty their contents in the cystine. Further is when we talk about the functions of the Golgi complex, what exactly the functions we have. Functions are nothing but is the Golgi complex is the secretory organ of the cell. It modifies, sorts and packs material synthesized in the cell and dispatches them to various targets like plasma membrane and the lysosome. It produces vacuoles and the secretory vesicles. It helps in the formation of the cell wall, plasma membrane and lysosomes. Next what we have is the lysosomes. Lysosomes are also called as the suicide bags of the cell. Why it is called as the suicide bags of the cell, that we will see in one of the functions of this particular lysosome. Lysosomes are simple single membrane bound sacs filled with the digestive enzymes. It contains the digestive enzymes. Those digestive enzymes are nothing but which helps in digesting a lot of things. Let's see the function of this, then it will be much clear. First of all, the immune system. It destroys viruses and bacteria that attack the cell. So it kills them. Demolition squads. It destroys worn out cellular organelles and organic debris, that is autolysis. Means if one of the cell is dead or died, or the cell organelle is died, then this particular cell organelle, it digests that or it destroys that, and that's why this is called as the autolysis. Next one, this is also called as the suicide bags. When a cell becomes old or is damaged, lysosomes burst and enzymes digest their own cells. During starvation, lysosomes digest stored proteins, fats. Further as next what we have to study is the mitochondria. This is one of the most important cell organelle of the cell. Mitochondria is also called as the powerhouse of the cell, because as we require electricity for our various machines to work exactly same way, when we talk about the cell to carry out their functions, they require energy, and that energy comes from mitochondria, and that's why this is also called as the powerhouse of the cell. Each cell, they have given also, each cell requires energy, and this energy is produced in the mitochondria. Under the electron microscope, a mitochondria is seen as double structure. Now when we talk about the mitochondria further, They have mentioned over here the outer membrane is porous and the inner membrane is deeply folded. These folds are called cristae. The inner cavity is filled with the proteinaceous gel-like matrix containing ribosomes, phosphate granules, and DNA. Therefore, it can produce its own protein with the help of enzymes, mitochondria, oxidizes carbohydrates and fats in the cell. The energy released in this process is stored in the mitochondria in the form of ATP, which we also call as the adenosine triphosphate. Plant cells have less mitochondria than animal cells because in the plant they require less energy, and that's why the number of mitochondria present in the plants are lesser as compared to the animals. Now, the functions when we talk about the produce or to produce energy rich compound called as the ATP, that is adenosine triphosphate, and synthesizes or the synthesis of proteins, that is carbohydrates, lipids, etc., by using the energy in ATP. Further is vacuoles. Vacuoles are also having their own importance. When we talk about the vacuoles, they are these storage sacs for solid or liquid contents. They don't have any typical size or shape. The structure of the vacuoles changes according to the need of the cell. Vacuole is bound by single membrane. Now the functions, the first one, to maintain the osmotic pressure of the cell. This helps in maintaining the osmotic pressure of the cell. To store metabolic byproducts and end products, that is glycogen, proteins, water, etc. In animal cell, they store waste products and food, while in amoeba, it stores food before digestion. And when the digestion is supposed to be taking place for the food, then the vacuoles are releasing them for the digestion. In plant cells, vacuoles are full of cell sap and provide turgidity or rigidity to them. The next what we have to study is the plastids. Plastids are also important for the plants. Actually, this one is present only in the plants. Why are plants leaves are green and flowers red, yellow, orange or blue? Means the different parts of the plants when we talk about, they have a different colors. So what makes or what gives them a different color? So that is nothing but what the plastids. Plastids have a double membrane, are of two types. So further, the different examples they have given, like a green color of the leaf is because of the chlorophyll, orange is because of the carotene, the yellow color is because of the xanthophyll, purple and the blue is because of the anthocyanin, and the dark pink is nothing but the betalains. Now the two types of the plastids what we have in that, the leucoplast and the chromoplast we have. The white or colorless plastids is called as the leucoplast and the colored plastids are called as the chromoplast. Chloroplast can get converted into other types of chromoplast. For example, raw green tomatoes turn red due to breakdown of the chlorophyll and synthesis of lycopene. There are so many examples, like if we talk about the raw mango, it's green in color, but as it gets ripened, it changes its color from green to the yellow color. Further as they have given about the functions for this, functions of the chloroplast. Chloroplast converts solar energy to chemical energy, that is food. Then the chromoplast give different colors to flowers and fruits. Leucoplasts are involved in the synthesis and storage of food like starch, oils, and proteins. Mitochondria and plastids can produce protein as well as replicate themselves, as they have their own DNA and ribosome. So this is one of the important point we have, that mitochondria and plastids can produce proteins as well as replicate themselves, as they have their own DNA and ribosomes. After studying the cell and cell organelles, we know exactly what the different parts we have, or what the different components we have inside the cell, which helps in carrying out the different functions of the cell. Another thing they have mentioned over here is that come to know that all the functions of the plant and animal cells are taking place smoothly due to the cell organelles. Such developed cells are called as the eukaryotic cells. Last year we have studied about the prokaryotic cells of bacteria. So now the comparison of both we can carry out. That comparison they have given at the end of the chapter. So eukaryotic cell, that is the size of 5 to 100 micrometer, whereas the prokaryotic cells, they are 1 to 10 micrometer. Means the developed cell, that is eukaryotic cells, they are bigger than the prokaryotic cells. In the number of chromosomes, more than one, only one in the prokaryotic cell. Nucleolus or the nucleus with the nuclear membrane, nucleolus, and the nucleoplasm, nucleide assembling nucleus, means a similar structure we had in the prokaryotic cell but not the developed one. Then the mitochondria and plastids present in the eukaryotic cells, whereas they don't have membrane bound cell organelles. So cell organelles were not there in the prokaryotic cell. And examples they have given over here is the present in highly evolved unicellular and multicellular plants and animals, whereas prokaryotic cells are present in bacteria. So this is all about the chapter cell and cell organelles.